Hi everyone, this is Yue. In this lecture, I will talk about RNA-seq. So first, I will talk about the biology and the experimental design in RNA sequencing. So what is transcriptome? The transcriptome refers to the set of all RNA molecules in one cell or a population of cells. In another word, it's a snapshot of expression profile of that cell or a group of cells at a particular time. So a transcriptome includes messenger RNA that encodes proteins. This should be familiar to you. Professor Li Juncheng already talked about it in her class, Microarray and RNA-seq for gene expression and differential expression analysis. But a transcriptome is not only mRNA. It can also include non-coding RNAs, such as microRNA, the blue one, and the long non-coding RNA, the green one. The long non-coding RNA is the non-coding RNA at least 200 base pair long. And it's a pretty hot area now. There are maybe more than 15,000 long non-coding RNA expressed in human genome, and it's just discovered in recent years. These RNA transcripts work all together to regulate many important physiological processes, such as cell growth, development, and apoptosis. As you are aware, there are many isoforms that are possible in most mammalian genes. This is a short summary of different kinds of splicing events that can occur. And the splicing events are often regulated by cis-regulatory sequences that live in introns. And this intron contain recognition sequence for splicing factors, where those splicing factors can be conditionally expressed. So you get different combinations of these exons being glued together to produce variant forms of proteins. So we have to be mindful of the idea that RNA molecules that we are going to observing typically are going to be reverse transcribed, and we will see entire transcripts that come most likely from distinct exonic locations in the genome. So to sequence RNA, the first thing we need to do is to isolate RNA species from a cell or a collection of cells in the desired condition. And note that we can choose which kind of RNA molecule to isolate. Within the nucleus, when the gene just transcribed, you get both the exon and intron. If you want to know the intron information or want to study, for example, long non-coding RNA, then you just get rid of the ribosomal RNA. Why is that? So in human cells, isolated total RNA, about 95% are ribosomal RNA. The remaining 2 to 3% are mRNA, and there are also 2 to 3% are long non-coding RNA or tRNA or microRNA. And the ribosomal RNA is very conserved throughout humans and is extremely stable in human tissue. In other words, measuring ribosomal RNA, the data cannot provide useful information. And mRNA is the most informative part of total RNA. That's why we want to remove ribosomal RNA, which are too abundant and will waste sequencing rates. Once the primary transcript become mature transcript, it will go to the cytoplasm for translation. So there you only get the exon, but no intron. There is a technique used oligo-T to only select the poly-A tail of mature mRNA to enrich exons, therefore get better signal. Of course, Some non-coding genes also contain a 3' poly A tail, which will also be selected. Most RNA-seq experiment is done this way. Only use the oligo T to select the poly A, the matured transcripts. Another area that need attention in RNA-seq lab preparation is strand specificity, or the directionality. We know that both strands of a DNA can be transcribed to different transcripts, the blue one or the red one. The commonly used Illumina RNA-seq kit is not strand specific, it's FR unstrand method. In other words, we cannot know which read of the pair is in the same direction as the transcript, and which one is complementary to the transcript in the opposite direction. For the stranded kit, there are two different scenarios. In the method, the FR first strand method, that is typically using DUTP labeling, or the true-seq stranded kit, or the Agilent sure-select strand-specific kit. In this method, the second read is in the same direction as the transcript. And this is kind of, I feel, is unintuitional, because it's called the FR first strand, but actually the second read is in the same direction as the transcript. And in the second strand method, the first read is in the same direction as the transcript. This method is usually used by the other platforms such as Solid. It's just the other way around. So the directionality is not that important for the well-known transcripts, but it could be very useful for novel genes, especially for the novel long non-coding RNA study. Here is a typical RNA-seq protocol. First, we get our RNA sample, and we remove the contaminated DNA from the RNA. And then we can select whether to remove two abandoned ribosomal RNA or just select the mRNA. And then we do the fragmentation. And then we reverse transcribe those RNA into cDNA. Then we can use either strand-specific RNA-seq kit or non-specific RNA sequence kit to ligate the sequence adapters to both ends of the fragments. And then we can decide whether use PCR amplification or not, depending on the initial amount of the RNA materials. And then we can select a range of the size. Typically, it's from 200 base pair to 400 base pair for the fragments. And then we just sequence the cDNA fragments. We can either do pair-end sequencing, like shown here, or we can just do single-end sequencing. And the sequencing technique is just the same as the DNA sequencing technique that I described in the NGS lecture. And the advantage of RNA-seq over microarray is that first is a digital representation. Every time you see a read, it's definitely some molecule comes from your sample. While for microarray, it's an analog signal. This region either has a high signal or low signal. It's not digital. And if you look at the Affymetrix array probe signals, there is a plateau toward the end when the transcripts become too abundant. The microarray in that spot will saturate the signal. In sequencing, the more you have some molecule, you will sequence more. And so RNA-seq has a higher detection range. Another advantage is that you don't need to know the transcript sequence. You just know for the isolated RNA sample, whatever it is, I will see it. You don't need to design microarray probes ahead of time. You can detect novel genes. You can study alternative splicing events. There are also some advanced applications. For example, in tumor sample, if a gene has mutation, then the transcript from that gene may also has a mutation. Also, you can study fusion genes. That is, there is one gene. There is another gene. Sometimes they are in different chromosomes, but somehow in cancer, they get joined together and they are transcribed. And in RNA-seq, you will see some read going from the first gene to the second gene. And also the cost is reasonable for both microarray and RNA-seq. And for microarray, it has relatively larger batch-to-batch variation compared to RNA-seq. And for experimental design, the sequencing part is just sequenced the same as in DNA sequence. And you can choose to do whether single-end sequence or pair-end sequencing. And nowadays, pair-end sequencing is very popular, and the price is quite similar. For the sequence steps, if you want to do differential expression analysis, 30 to 50 million reads is good enough. And if you want to do transcript assembly or splicing event study, you need 100 million reads. And for the read length, typically it's 75 base pair to 150 base pair. If you want to do transcript assemble or splicing event study or mutation call, then I would recommend longer reads. And if you want to do expression profile, then for the same throughput, I would recommend shorter read length, but more number of reads. For replicates, still, even though a next generation sequencing, you can get rid of the technical replicates, but you still need biological replicates, and the more the better, at least 3 for publication. Batch effect still exists. Still try to be consistent and process all the samples at the same time in the same lane if possible. Please remember, better technology never eliminates the needs for good experimental design.